ECC, or otherwise known as excitation-contraction coupling, refers to the sequence of events by which an electrical event in the sarcolemma of the muscle cell initiates the sliding of the myofilaments, resulting in contraction. The process of ECC links the electric excitation of the surface membrane to contraction. There are 10 steps to the excitation-contraction coupling process. Step 1 is when the AP arrives at axon terminal at neuromuscular junction. Step 2 is when the ACH is released, it binds to receptors on sarcolemma and action potential reaches the T-tubule. Step 3 is when the sarcoplasmic reticulum releases calcium. Step 4 is when the active site exposure and the cross-bridge binding. Step 5 is when the contraction begins. Step 6 is when the ACH is removed by the ACHE. Step 7 is when the sarcoplasmic reticulum recaptures the calcium chloride. Step 8 is when the active site's covered and no cross-bridge interaction is happening. Step 9 is when the contraction ends, and step 10 is when relaxation occurs and passive returns to passive length.
Excitation-contraction coupling is the mechanism by which muscle fibers translate an electrical stimulus into a mechanical response. This is how muscles contract. As a nerve impulse reaches the end of an axon terminal, it stimulates the release of acetylcholine from synaptic vessels. Acetylcholine then diffuses across the synaptic cleft and attaches to the acetylcholine receptors on the sarcolemma at the neuromuscular junction. After acetylcholine attaches to its receptors on the sarcolemma, the muscle fibers become more positively charged. As the action potential travels along the T-tubules, calcium is released from the sarcoplasmic reticulum. Calcium binds to troponin on the actin filaments of the muscle fibers. This signals tropomyosin to move out of the myosin-binding sites on actin. Energized myosin heads with an attached ATP molecule and phosphate group bind to the open myosin-binding sites on actin. This forms an actin-myosin cross-bridge. After the cross-bridge forms, the myosin head rotates towards the center of the sarcomere. As it rotates, the myosin releases its ADP and phosphate. This action slides with the thin actin past the thick filament, creating the power stroke. After the power stroke, the actin-myosin cross-bridge remains intact until an ATP molecule binds to the myosin head. After ATP attaches to the myosin detaches from the actin.
SFT is the sliding filament theory. It describes the mechanism that allows muscles to contract. According to this theory, the myosin binds to actin. The myosin then alters its configuration, resulting in a stroke that pulls on the actin filament and causes it to slide across the myosin filament. On this slide shows the sliding filament theory. Step 1. Exposed binding sites on actin allow the muscle contraction cycle to occur. Step 2. Cross-bridge binds actin to myosin. Step 3. Cross-bridge pulls actin filament, ADP and P released from myosin. Step 4. New ATP binds to myosin, causing linkage to release. Step 5. ATP splits, which provides power to cock the myosin cross-bridge. On this slide, it shows the sliding filament theory going into more depth and how this cycle works.
Okay, so let's have a closer look so that we can see the details of how this works. The pink molecules that you can see on screen are myosin, which at rest have both ADP and phosphate molecules attached to the myosin heads. The other molecules that you see are actin, which is in light blue, tropomyosin, which is in dark blue, and troponin, which is in green. When we decide to move, calcium is released from the sarcoplasmic reticulum. These molecules bind with the troponin molecules, which cause the troponin-tropomyosin molecules to move, revealing myosin attachment sites on the actin myofilaments. The single phosphate attached to the myosin heads gets released as the heads move to connect to the attachment sites on the actin. This formation is called a cross bridge. The remaining ADP molecule is then extended in what is called a power stroke. and this occurs when the myosin heads fully act inward. Once the ADP attachment is spent, the ATP molecules attach themselves to the myosin heads. These molecules trigger the release of myosin heads from the actin attachment sites and are immediately broken down by ADP and a single phosphate as the heads move back into the resting position, hence being in the recovery stroke that you see now. If calcium is still present, the cycle will repeat again and the sarcomere will continue to shorten until the calcium ions are transferred back into the sarcoplasmic reticulum, as you can see.